I'm Dr. Shrikant Chetta, the Pharmacovigilance Guru, and welcome to my channel PV Signal Guru, tailored for pharmaceutical professionals and organizations. This channel is your go to resource for mastering the intricacies of pharmacovigilance. With expertly curated content rooted in GBP modules and global regulatory guidelines, PV Signal Guru equips you with the knowledge and tools to confidently navigate the ever evolving landscape of drug safety and compliance. The training sessions are designed to simplify complex pharmacovigilance concepts while delivering practical insights, ensuring you stay ahead in this vital field. Join PV Signal Guru to enhance your expertise and drive excellence in pharmacovigilance. Thank you for tuning into the first of many insightful sessions on pharmacovigilance. In today's episode, we will dive into the basics of signal in pharmacovigilance. Whether you are new to the field or looking to refresh your knowledge, this session will provide a clear and foundational understanding of this critical concept. Let's get started. In this session, we'll cover the following key topics. What is a signal? What are the sources of a signal? Signal sources that align with ICSRs, that is individual case safety reports. Sections of a PBRER aligning with signal sources. Signal management, an overview of the process. The evolution of a signal. Activities impacted by newly identified signal. And the importance of signal process. By the end of this session, you will have a solid understanding of the basic principles of signal in pharmacovigilance and be well equipped to apply this knowledge in your work. Let's dive in. What is a signal? According to the Sioms group, a signal is defined as information that arises from one or multiple sources, including observations and experiments, which suggests a new potentially causal association or a new aspect of a known association between an intervention and an event or a set of related events, either adverse or beneficial, that is judged to be of sufficient likelihood to justify verificatory action. To qualify an adverse event as a signal, there are four key elements: an intervention and an event. An intervention refers to a specific treatment, procedure, or action intended to improve health or well-being. This could include a drug or a medication, a medical device, a procedure, behavioral changes, diagnostic strategies, treatment strategies, or a prevention strategy. An event, from signal perspective, can have adverse effect or a beneficial effect. However, from safety aspect, signal is usually considered to be an adverse event. In addition to intervention and event, we need to have a new association, an event with a product which was not known earlier, or a new aspect of a known association. That is, a new characteristic of a known adverse event. And these justify a verificatory action. In simpler terms, a signal is identified when new information about a product raises enough concern to warrant further investigation. It's essentially a red flag that prompts us to dig deeper into potential safety concerns or benefits. Now, let's explore what qualifies as new association or a new aspect of a known association. A new association can include a new previously unlisted adverse event. For example, a widely used antidepressant is found to cause acute pancreatitis, a side effect not previously documented in its safety profile. This new adverse event may lead to updates in the drug's labeling, including warnings and precautions for patients and healthcare providers. It could also be a new drug-drug interaction. For example, a common blood thinner such as warfarin, which is known to cause many drug-drug interactions, is discovered to interact with a newly approved antibiotic, leading to an increased risk of severe bleeding. This interaction may require updates to prescribing guidelines, contraindications, or monitoring requirements for patients on both these medications. It can be a new drug food interaction. For example, a medication used to treat high blood pressure is found to have reduced efficacy when taken along with grapefruit juice due to interference with the drug metabolism. This interaction may prompt updates to the drug labeling, advising patients to avoid grapefruit juice while on medication. A new drug alcohol interaction, a new psychotic drug, is found to cause dizziness and drowsiness when taken with alcohol, increasing the risk of falls or accidents. This interaction may lead to warnings about alcohol consumption while on medication as well as patient education material. And finally, it can also be a new beneficial effect. A drug originally approved for treating diabetes is found to have beneficial effect on reducing the risk of heart attacks in diabetic patients. This new beneficial effect may lead to expanded indications for the drug, updating labeling, and new marketing strategies to highlight the additional benefits. Now, let's look at the new aspects of known association. It could be increased frequency. For example, a known side effect of a drug is headache, which was reported in 5% of patients. However, over time, pharmacovigilance data shows that the frequency of headaches has increased to 15% in the same patient population. This change in frequency signals a potential change in the drug safety profile, prompting further investigations into the factors like dosage, patient demographics, drug interactions, or prescribing strategies. And a new aspect can also be increased severity of an event. For example, a drug is known to cause mild skin rashes in some patients. However, recent reports indicate that some patients are now experiencing severe rashes, such as Stevens-Johnson syndrome, SJS, which is a life-threatening condition. This escalation in severity requires immediate attention, including label updates and risk minimization measures. A new aspect can also be a new population at risk. For example, a drug used to treat hypertension is known to cause dizziness in elderly patients. The new data reveals that young adults are also experiencing dizziness at a higher rate than previously observed. This signals a need to reassess the drug safety in younger populations and potential update on the prescribing guidelines. It could also be a new outcome related to an event. Example, a drug associated with liver enzyme elevation, which is a known event, is now linked to acute liver failure and hospitalization in some patients, which is a previously unreported outcome. This new outcome may require additional warnings, monitoring, or even restrictions on the drug's usage. And finally, there could be an increased specificity in identifying the event. For example, a drug is known to cause gastrointestinal, that is GI discomfort, and recent reports suggest that it is very specific and the discomfort is primarily due to severe gastritis, which is nothing but inflammation of the gastric or the stomach lining, rather than general GI issues. So this increased specificity helps us refine the drug safety profile and may lead to targeted risk management strategies, such as recommending proton pump inhibitors for at-risk patients. As we see, new associations and new aspects of a known association in pharmacovigilance can significantly impact drug safety profiles, regulatory decisions, and patient care. Identifying and addressing these associations is crucial for ensuring the safe and effective use of medicinal products. Identifying these associations help us determine whether information qualifies as a signal, prompting further investigation to ensure patient safety and regulatory compliance. As we learned from definition of a signal, it can arise from one or multiple sources. Let's explore the diverse range of sources where a signal may originate. ICSRs, individual case safety reports. These are reports of adverse drug reactions or other safety related information for individual patients. They are submitted to regulatory authorities and serve as a primary source of safety data. Periodic reports. These include documents like the periodic safety update reports PSURs and periodic benefit risk evaluation reports PBRERs, which summarize the safety profile of a drug over a specific period and are routinely submitted to regulatory authorities. Medical and scientific literature, published studies, case reports, and reviews in medical and scientific journals are critical sources of safety signals. Pharmacovigilance teams regularly review this literature to identify potential safety concerns. Regulatory authorities, agencies such as FDA, EMA, and MHRA play a central role in pharmacovigilance. They collect and analyze safety data, issue guidelines, and may mandate actions like label updates, risk minimization measures, or protocol withdrawal or product withdrawals. Regulatory submissions, PSURs, RMPs, and as well as variations as well. So changes to drugs marketing authorization such as updates to the product information or manufacturing processes. Drug information centers. These centers provide information on drug use and may also report ADRs. Patients. Patients can directly report ADRs through patient support programs or through online portals or through a phone line. Media. News reports or social media discussions may highlight potential safety concerns as well. Poison units. Information from these units provide very important data on drug overdoses or misuse. Sales representatives. They may receive feedback from healthcare professionals regarding adverse events, which are reported back to the marketing authorization holders. Legal personnel. Safety issues may also be identified during litigation or legal reviews. Directly from the healthcare professionals, doctors, pharmacists, and nurses, these are the primary sources of ADR reports. Clinical trials. Safety data from ongoing or completed clinical trials can also provide information on new risks or trends. They may also come from business partners, and we also receive it from pharmaceutical manufacturers. Companies are also responsible for monitoring and reporting safety data. And finally, epidemiological studies. Population-based studies can identify trends or associations between drugs and adverse outcomes. These diverse sources collectively contribute to the identification and evaluation of signals, ensuring a comprehensive approach to drug safety monitoring. The list of signal sources is indeed extensive, but there are simpler ways to remember them, whether you are new to pharmacovigilance with a focus on ICSRs or an experienced professional working on aggregate reports like PSURs or PBRERs. Let's break it down in a way that's easy to grasp and apply. Let's explore the sources of signals that align with ICSRs. In pharmacovigilance, any new data or even any event reported for a medicinal product by a marketing authorization holder eventually finds its way into the company's safety database. Therefore, the sources used to create ICSRs are also the primary sources of signals, As you may know, ICSR are categorized into two types based on the sources: spontaneous cases and solicited cases. Let's look at the spontaneous sources. A spontaneous report is an unsolicited communication from a healthcare professional, consumer, or other stakeholders to a regulatory authority, MAH, or organization. It describes one or more suspected adverse reaction in a patient who was given one or more medicinal products. These reports do not originate from studies or organized data collection system. Some of the sources would be healthcare professionals, example doctors, pharmacists, nurses, literature, consumers or patients reporting through patient support programs, online portals, or through a phone line. Pharmaceutical manufacturers or marketing authorization holders, regulatory authorities like FDA, EMA, MHRA, business partners like the licensees or collaborators, World Health Organization, and poison control centers. Now let's look at solicited cases. Solicited reports are derived from organized data collection systems, and these include clinical trials, safety data coming from ongoing or completed trials, registries, patient registries tracking specific diseases or treatments, named patient use programs, such as early access programs for unapproved drugs, patient support programs, programs providing assistance to patients using specific medications, disease management programs, initiatives focused on managing chronic conditions, surveys of patients or healthcare professionals, gathering feedback on drug use and safety, and post-authorization safety studies conducted after a study is approved to monitor the long-term safety. Now that we've seen how signal sources align with the types and sources of ICSRs, it's clear that high quality ICSRs play a crucial role in pharmacovigilance. Accurate and detailed ICSRs are the foundation for identifying and evaluating potential safety concerns, making them essential for ensuring patient safety and effective drug monitoring. By understanding these sources, you can better identify and evaluate signals, whether you are working with spontaneous reports or organized data collection system. This knowledge is essential for ensuring robust pharmacovigilance practices. Just as signals from all sources are captured in pharmacovigilance through ICSRs, signals identified during the review period of a PSUR, periodic safety update report, or a PBRER, periodic benefit risk evaluation report, are also represented in these documents. Additionally, the various sections of a PBRER can reflect the sources of signals. Let's explore how the sections of a PBRER align with signal sources. Section 6, which is data and summary tabulations. While this section doesn't focus on a specific source, it reflects the cases included in the company's safety database, whether spontaneous or solicited. It provides the number of cases or events received during the review period and cumulative counts. The sources would be spontaneous or solicited cases for patients, HCPs, legal personnel, or sales representatives. As we've seen earlier, all the sources for ICSR creation. Section 7, which is summaries of significant findings from clinical trials. This section focuses on clinical trials, which are a key source of signals. So during ongoing or completed trials, any new safety information coming in would also trigger a signal. Section 8, findings from non-interventional studies. This section includes data from marketing authorization holder sponsored non-interventional studies such as registries, patient support programs, observational studies, or epidemiological studies, all of which are potential sources of signals. Section 9, information from other clinical trials and sources. This section covers information from business partners or investigator-initiated trials, which are also the sources of signals. Section 10, non-clinical data. This section includes findings from the preclinical or the non-clinical studies. Section 11, literature. This section reviews medical and scientific literature, which are a critical source of signals. Section 12, other periodic reports. This section includes other PBRERs prepared by the marketing authorization holder for different indications or fixed combination products. And finally, section 15, which is overview of signals, new, ongoing, or close. In this section, health authority requests to evaluate safety topics are presented, which can be considered as source of signals originating from regulatory bodies. By understanding how these sections of a PBRER or a PSUR align with signal sources, you can better navigate the process of identifying and evaluating signals during the preparation of periodic reports, ensuring consistency in safety data presentation. This structured approach ensures comprehensive pharmacovigilance and robust drug safety monitoring. Now that we've explored what a signal is, its various sources, and how these sources align with both ICSRs and PBRERs, it's time to dive into the signal management process. This process is a critical component of pharmacovigilance, ensuring that potential safety concerns are identified, evaluated, and acted upon effectively. According to GBP Module 9, the signal management process is defined as a set of activities performed to determine whether, based on an examination of ICSRs, aggregated data from active surveillance systems or studies, scientific literature information, or other data sources, there are new risks associated with an active substance or a medicinal product, or whether known risks have changed, as well as any related recommendations, decisions, communications, and tracking. The EU signal management process consists of the following key activities like signal detection, signal validation, signal analysis and prioritization, signal assessment, recommendation for action, communication. Let's break down each step. Signal detection. This is the process of identifying potential signals using data from various sources. The data sources could be spontaneous reports, clinical trials, literature, or observational studies, or real-world evidence, etc. All the sources that we learned earlier. And the methods that we follow to perform signal detection could be quantitative, that is disproportionality analysis, or qualitative methods, or a combination of both. The goal over here is to identify unusual patterns or trends that may indicate a new or changed risk. Next is the signal validation. Over here, we try to identify whether the signal is new or previously known. The steps involves assessing the quality and reliability of the data supporting the signals. And what we focus is to ensure the data is, as I said, credible and warrants further investigation. Once a signal is validated, we perform signal analysis and prioritization. Here, the strength of evidence, clinical relevance, and potential public health impacts are evaluated. The factors considered are the severity, the frequency, and impact on patients. And the outcome, what we are trying to see over here is, based on the available information, how do we prioritize the signal, and then what is the urgency on the actions that we need to take. And following the analysis and prioritization, one thing what we need to be aware is prioritization happens throughout the signal management process. It's not just after validation. It's a continuous process. And signal assessment. This is a thorough evaluation of the signal to determine if it represents a new or changed risk. The data we analyze would be review of clinical trials, epidemiological studies, and all other relevant data. A thorough, detailed evaluation looking at the strengths and evidence of each and every source is performed to say whether it is a new risk or a changed risk. The consideration what we look into is the causality, the severity, the population affected, and the overall risk benefit profile of the product. So at the end of signal assessment, we clearly document the findings from various sources, evaluating the strengths and weakness, and finally making the conclusions to either refute the signal or confirm it as a risk. And based on this assessment, we propose the appropriate recommendations for actions. Again, the recommendations could be updating the product label or the RMP, or conducting the further studies, or implementing any risk minimization measures. And the recommendations what we propose are usually submitted as a variations to the regulators as well. And the actions that we propose are proportionate to the risk which is identified. And finally, for the risks that we have identified, we need to communicate to the various stakeholders. The stakeholders could be regulatory authorities, healthcare professionals, patients, internal teams, or business partners. The goal over here is to ensure transparency and informed decision making for all the stakeholders. So, from the overall signal management process, what are the key objectives and the key questions that needs to be addressed at each and every step of this signal management process, from detection to communications? So in signal detection, what we are trying to say is there any new information coming from review of various sources for our product? In validation, what we are trying to say is this signal related to the product, and does it justify performing a deeper analysis? When it comes to analysis and prioritization, what we are trying to say is, okay, if this could be a signal with a product, what could be the effect of it, and what could be the perceived impact of it, and how do we react it? Sometimes, as I said, prioritization does not happen after validation, it happens throughout the process. So sometimes we need to take immediate actions at the detection phase itself and not wait till the final evaluation is completed. So over here, analysis and prioritization, it also helps us to take or react. How fast do we need to react it? Do we characterize a new signal to be a standard risk or a high impact risk? When it comes to assessment, what we're trying to find is we are doing a comprehensive evaluation of all the data sources, and we are documenting the strengths and the weakness of each source to come to a final scientific conclusion whether it is a confirmed risk or do we refute it. So everything is documented in a signal evaluation report or assessment report. This is where we actually document the complete thorough evaluation. And the recommendations for action. Okay, as per the assessment, we have assessed and we confirm it to be a risk. So now that we know that our drug causes an event, how do we minimize this risk? As a marketing authorization, it is our responsibility to ensure that the benefits always outweigh the risk. And whenever there are risks identified, we need to propose the appropriate risk minimization measures. And that is what we do in the recommendations for actions, focusing on minimizing the risk which is newly identified. And finally, the communication is assessing who all needs to be informed so that they can take informed decision. By following the structured signal management process, pharmacovigilance teams can effectively identify, evaluate, and address potential risks, ensuring the safety of medicinal products and protecting the public health. So the overall key takeaways of the signal management process is that it is a structured, step-by-step process designed to safeguard patient safety. Each step from detection to action and communication builds on the previous one, ensuring a thorough evaluation of risks. And it also tells us that collaboration and communications are critical to the success of signal management, ensuring all stakeholders are informed and are aligned. This signal management process is a very deep subject, and in our upcoming sessions, we'll dive deeper into the various steps of signal management process. So stay tuned for detailed insights, and if you found this video helpful, don't forget to like and subscribe to get notified about our latest updates and new content on this. The information reported in a signal is not conclusive and can change significantly over time as more data becomes available. The evolution of a signal in pharmacovigilance involves several stages, starting from the initial observation of a safety concern to its classification as a potential or confirmed risk. This process is crucial for ensuring patient safety and maintaining the benefit-risk balance of medicinal products. So now let's explore the stages involved in signal evaluation. Signal detection. A potential safety concern for a drug event pair is identified by reviewing various sources, and this sparks interest in performing verification action to validate the signal. The signal validation happens over here. So the new information for the drug event pair is evaluated for clinical relevance, previous awareness, and supporting evidence from other sources. Based on this evaluation, the signal is either validated or non-validated if it is not supported by sufficient evidence. Understanding this evaluation helps pharmacovigilance professionals effectively monitor and address safety concerns, ensuring the continued safety of medicinal products. So, what is a validated signal? So, a signal for which the signal validation process has verified that the available documentation contains sufficient evidence demonstrating the existence of a new potentially causal association or a new aspect of a known association, and therefore justifies further analysis of signal. And a non-validated signal is something which, after the signal validation process, has led to the conclusion that the available documentation at this point of time does not contain sufficient evidence demonstrating the existence of a new potential causal association or a new aspect of a known association, and therefore further analysis of the signal is not warranted, and hence for a non-validated signal there is no further process of evaluation, but for a validated signal we perform the analysis. As I said, a validated signal is subjected to further evaluation where all available evidence is thoroughly reviewed. The goal is to determine whether there are new risks causally linked to the active substance or medicinal product, or if known risks have changed. This comprehensive review may include non-clinical data like preclinical studies, clinical data, trials, real-world evidence, or other sources of information. At the end of the signal assessment, the signal may be refuted if the evidence does not support a causal link, or confirmed if the evidence supports a causal link leading to its classification as a risk. This step ensures that only well-supported risks are acted upon, maintaining the integrity of pharmacovigilance practices. A signal that is confirmed as a risk is further categorized based on its impact on the benefit-risk balance of the medicinal product. It is characterized either as a risk not characterized as important if the risk has minimal impact on the overall benefit-risk profile, or important risk if the risk significantly affects the benefit-risk balance and requires attention. This classification helps prioritize risks and guides appropriate regulatory and safety actions. Based on the strength and evidence of the causality, a confirmed risk is further classified as either identified risk or a potential risk. For any confirmed signal, the minimum risk minimization activity that involves is updating the reference safety information and the associated labels such as USPI or SMPC or any country specific labels. Additionally, for risks characterized as important risks, update to the risk management plan and the proposal of additional risk minimization measures may be required. It's important to know that the RMP focuses only on important risks, specifically important identified risks, important potential risks, and missing information. So risks not characterized as important are not included in the RMP, but are still addressed through updates to the RSA and product labels. This structured approach ensures that the risks are managed appropriately based on their significance and impact on patient safety. So, the classification of a risk depends on two key factors. One is the strength of evidence supporting the new association and the impact of this association on patient and public health. These factors guide the development of effective and practical risk minimization measures, ensuring that the actions taken are both efficient and implementable to safeguard patient safety. Now that we've explored what a signal is, its sources, the signal management process, and signal evolution, let's dive into understanding the impact of a signal on a product. A confirmed signal can influence various aspects of a drug's life cycle, including drug development or the product development. If the product is in ongoing clinical trials, the new safety information may impact its development. This could involve updating the investigator's brochure, IB, partial or complete suspension of the trial, recall of the investigational drug, or protocol modifications. It can also lead to label updates. The new safety information must be reflected in the company's reference safety information RSI and other labels such as USPI, SMPC, or other country specific labels. Sections that may be updated include the undesirable effects, warnings and precautions, contraindications, drug interactions, use in pregnancy and lactation, overdose, and other sections as applicable. Risk management. Depending on the impact of the newly identified risk, appropriate risk minimization measures are proposed and implemented. These may include updating the RSI or RMP, planning a past study, recall or withdrawal of the product, implementing restricted access programs, introducing patient alert cards or prescriber guides, additional monitoring, and many more. The signal can even impact the product surveillance strategy. The evolving safety profile may require changes to the product surveillance strategy, such as adjusting the sources to be reviewed or the frequency of review. It may also trigger keeping some events under close monitoring. A signal would also impact the aggregate safety reports, such as if all validated signals, regardless of the status, are included in PSURs or PBRERs. Updated safety information may alter the benefit risk balance, impacting sections such as Section 4, changes to the RSI, Section 15 and 16, including risk characterization, Section 18.2, benefit risk evaluation, the conclusion executive summary, and Appendix 3's signal table. In addition to that, the sources of signal and the section would also be impacted. By addressing these areas, the impact of a signal ensures that the drug safety profile remains up to date, protecting patient health and maintaining regulatory compliance. In our upcoming sessions, we'll explore the sections impacted by PSURs or PBRERs and discuss various scenarios in detail. If you found this video helpful, don't forget to like and subscribe to stay updated with our latest content. Timely identification of signals is a cornerstone of pharmacovigilance, playing a critical role in ensuring drug safety and public health. Here's why it matters. Patient safety. Early detection of potential risks allows for swift action to protect patients. This may include updating product labels, issuing safety communications, or in extreme cases, withdrawing the drug from the market. Regulatory compliance. Regulatory authorities like the FDA and EMA mandate continuous monitoring and reporting of safety signals as part of post marketing surveillance. Timely identification ensures compliance with these requirements. Risk management. Early signal detection enables the implementation of risk minimization measures such as risk evaluation and mitigation strategies or additional risk minimization measures to ensure the safe use of the drug. Public trust. Proactive signal management demonstrates a commitment to patient safety, helping to maintain trust in healthcare systems and pharmaceutical companies. Economic impact. Delayed identification of safety issues can lead to costly consequences, such as litigation, drug recalls, and loss of market share. Early detection helps avoid these financial risks. Scientific understanding. Timely signal detection enhances the overall understanding of the drug safety and efficacy, guiding future research and development efforts. As we wrap up today's session on the basics of PV signal, we've come a long way from asking, what is a signal in pharmacovigilance, to graduating with a solid understanding of the basic principles of signal in pharmacovigilance. Here's a quick recap of what we covered. Understanding the definition and significance of a signal in drug safety. Exploring where signals originate from ICSRs to literature and beyond as sources of signal. Making it easier to identify signals in ICSRs by going through the solicited and spontaneous case reports. Connecting signals and their sources to various sections of aggregate reports. A brief overview of the structured steps to detect, validate, and manage signals as part of signal management process. And then we looked at the signal evolution by tracing how new safety information evolves from a signal to a confirmed risk. And we also learned what is the impact of a signal by understanding how a newly identified signal can influence product development, risk management, label updates, safety surveillance, and aggregate reports. And finally, we also learned the importance of the signal process by recognizing why timely signal detection and management are critical for patient safety, regulatory compliance, and public trust. Thank you for joining this session. If you found this content valuable and would like to explore more aspects of pharmacovigilance, please like, share, and subscribe to stay updated with our latest videos. Stay tuned for our upcoming sessions where we'll dive even deeper into these topics. If there are specific topics you would like us to cover, feel free to reach out at guru@pvsignal.guru. Signing off for now, this is your PV Signal Guru, Dr. Srikant Chetta. Thank you for watching, and I look forward to seeing you in the next session.